All right, guys, welcome back to Underground Science. In this video, we'll be talking about the last type for the complement pathway, and this is going to be the lectin pathway. Okay, so why is this pathway important? Well, certain pathogens, right, have mannose on them. Okay, and if you have mannose on them, we need a way to get rid of these pathogens also. And it turns out our body does produce something called mannose binding lectin. Okay, so there are certain pathogens that have mannose on them, right? Certain pathogens that have mannose on them, and our body produces something called mannose binding lectin, right? So let's say it's represented like this. Our body produces mannose binding lectin, right? And of course, with the name, you can tell what it does, right? Mannose binding lectin binds mannose on pathogens, right? That have mannose on them. Okay, so let's see how this lectin pathway works. So let's say we have a pathogen, and this pathogen has mannose on it, right? Our body produces mannose binding lectin that goes and binds the mannose on the pathogen. So let's represent that like this, right? So this is our mannose binding lectin, and then we have our mannose here in white, which is present on the pathogen, right? It's a sugar. Then once that happens, right, once our mannose binding lectin binds to the mannose on the pathogen, this then attracts two other main molecules that we have to talk about. All right, so this leads to the activation of MASP1 and MASP2. Okay, they stand for mannose binding lectin associated serine proteases. All right, so that's why we're just going to abbreviate it. So what happens next? This attracts two different molecules called MASP1 and MASP2. All right, so MASP1 and MASP2. Okay, so this would be MASP1 and this will be MASP2. All right. Again, this stands for, we'll just write it right here, right? Mannose binding lectin, mannose binding lectin associated serine protease. All right. So we're gonna have MASP1 and MASP2. That's gonna come and bind once mannose binding lectin binds the mannose on the pathogen. All right. Now MASP2 will be the one that cleaves different things, just like we saw in the classical pathway. C1s was the one that cleaved C4 and C2. Just like that, MASP2 will cleave C4 and 2, C4b and C4a, right? And then what's that going to lead to? C4a, we know, will is important for inducing, or C4b is going to be the one to bind, but we know C4a does what? Induces mild inflammation, right? We talked about this when we did the classical pathway. C4b is going to be the complement protein that actually binds the microbial surface. So I'm going to represent that right here. All right, so this is going to be C4b. Next, MASP2, just like C1s, will also cleave C2 into two different things. What are those two different things? C2a and C2b. We know that C2a will go and bind C4b. We'll draw that in green. So let's do a different color. Let's do um, red, right? This will be C2a. Okay, we know that C2a will go and bind C4b. Remember from our complement system video, what does C4b2a make up, right? So what is C4b2a? This is C3 convertase, okay? And what does C3 convertase do? It takes C3 and converts it into C3a and C3b, right? We know that C3b can go and directly bind onto pathogens because it's a potent opsonin. So it can go bind to pathogens, and then neutrophils and macrophages can go and destroy the pathogen, right? But also we know that C3b can go and bind onto C3 convertase, in this case C4b2a. What does that lead to? So if we have our pathogen here, we have our C4b, our C2a, and then our C3b that goes and binds. What is this now? This is your C4b2a 3b complex. What is C4b2a 3b? This is now called C5 convertase. What does C5 convertase do? It converts C5 into what? C5a, C5b, right? We know C5a induces inflammation and attracts and activates neutrophils. And if we actually take a moment here to go back to C3a, remember C3a activates histamines and induces inflammation also. So let's not forget that because it helps attract white blood cells to the area of infection. So anyways, back to here. So we saw C5a, what it does. What does C5b do? Now remember, C5b is what starts the formation of the membrane attack complex. So C5b will then bind, it's not C56, C5b will then bind onto C6, C6 will bind onto C7, C7 will bind onto C8, C8 will bind onto C9, and this whole thing is going to form a membrane attack complex, right? So this will form a membrane attack complex onto where? Onto the pathogen. So if we come here, we'll form a membrane attack complex, which is going to create a pore, right? That's going to help water from the outside environment to flow inside the pathogen, which will lead to osmotic lysis and cell destruction, okay, and pathogen destruction. So that's a lectin pathway. It starts directly from C4, right, after MASP2 cleaves it. All right, and that's basically it. That's the lectin pathway. And I hope this video made sense. I hope it wasn't too confusing. Thanks for watching, and see you guys in my future videos.